Day 2 of our GMO lab uses one cool tool, the micropipette, and a really awesome biotech technique called polymerase chain reaction. Before you watch this video, be sure to familiarize yourself with how to use a micropipette and what PCR is. In this lab specifically, we're going to set up our lab with two different primers, one that's going to identify just a housekeeping plant gene. That's a gene that is pretty much found in every cell. It's active, it's turned on. So we're looking for a regular old plant gene and a genetically modified organism gene. So that's that promoter and terminator region that we talked about in the first video. We're going to have three sources of DNA. Two we prepared in the last lab, that's our test food sample and our GMO negative sample. So that's a negative control, so we know that those oats were not from genetically modified genes or should not have genetically modified genes in them. And today you're going to get a positive control, so just in case your DNA extraction didn't go so well, you're going to get purified DNA in the form of a GMO positive. So you really should get to see how the plant primers are going to turn out and how the GMO primers are going to turn out with PCR with this GMO positive sample. So 2 different primers must be mixed with 3 different DNA samples. That means we're going to need 6 PCR tubes that we're setting up per group. So here they are, our 6 tubes. You're going to label them because there's not enough room on the tubes to actually write what's going in them, but everything's going to look pretty similar, so be sure to write numbers on the top of the lids. Also, write your group number because everybody's numbers are going to be the same, so I would add group numbers to the beginning of those numbers. And then, if you look at what's going inside, into all 6 tubes is 20 microliters of master mix. 3 of those are going to be plant. So the plant genes, the primers for the plant genes are going to be mixed in with that master mix, and the primers to identify the genetically modified genes, those are going to be mixed into the red sample. So the only thing that's different in these master mixes are the primers. You're still going to have buffers and nucleotides and polymerases, the things you'd find in any PCR reaction. Now we're ready for our 3 DNA samples. So once the master mixes are in, you'll add your 20 microliters of non-GMO control to the first 2. So that is your oats. So that should come out of an InstaGene matrix tube. And then you've got your test food. That's also going to come out of an InstaGene matrix tube. And then 5 and 6, you're going to add DNA from the tube I give you today, and that's the GMO positive control. So you're getting 3 things from me. You're getting a green master mix, a red master mix, and a clear tube that's got GMO positive DNA in it. Now, when you start to pipette in these liquids, contamination is a really big factor now, because if you mix test food or positive control in with another, you're through PCR going to amplify that contaminant as well as whatever is supposed to be in the tube. So when you're putting in the master mixes, you're pipetting into sterile tubes. So it is okay with me if you use one pipette to put in all the plant primer master mixes, and then another, please eject your pipette tip and get a new pipette tip to put in all your GMO primers. Then you have to use a separate tip every time to enter the other 6, and so I want you to close the lids as you put in your DNA from your non-GMO control, from your test food, and from your GMO positive control. And you need to change your tip every time, because even contamination from the primers going into a different vial could cause problems. All right, so now you know the trick. As you put in that DNA, close that cap so that you know you've done it and you don't get confused. But here's just a little bit of background on how removing liquid and adding liquid to these tiny tubes goes. So when you're removing liquid, your micropipette is straight up and down, and the tip is not touching the bottom of the vial necessarily. Ideally, it would be floating, like kind of hovering below the surface of the liquid, but not quite touching the bottom. When you go to add liquid, you actually touch the pipette tip to the side of the PCR tube or the microcentrifuge tube, and that gets you like optimal, removal of the liquid from the pipette tip. So that's at an angle, touching the side. So those are just a couple of tricks to make your micropipetting go a little bit smoother. But you're dealing with very tiny volumes, so don't be worried if, it seems like you should have more liquid left in the vial you're pipetting out of and you run out. Like, things happen. Just be careful. And that's the basics for today. Hope you have fun.